Hey there, everybody. Thank you again for joining us. We have a great session for you today on understanding startup valuation and all about SMART Cap tables. If you have any questions during the session, you can go ahead and put them in our chat box, which I'm unlocking right now. And with that, I'm going to hand it off to Hall, our CEO, to get things started.
Great. Thanks, Shane. And want to thank everybody for joining us today. We have a great presentation. Today we have Christian Apfel, who's going to be talking about cap tables and how to calculate cap tables with the Smart Value cap table tool that he has. And I had the pleasure of having Christian on my podcast the other day and found he has a wealth of knowledge about this. He's an experienced life science investor. He has a long track record of successful startups and as well as investing, and invited Chris to come on today and share that with us and talk about it. So with that, we're going to pass it over to Chris, and then afterwards we'll have two presentations from companies pitching. So with that, Chris, or Dr. Apfel, let's go ahead and kick off. Let's tell us more about yourself and what you're doing there.
Well, thank you for having me here a second time. It's my real pleasure. I'm Dr. Chris Apfel. I have a couple of letters after my name. That's all. My excuse is to get properly through life. I needed more and more education. As you will see, it actually helps if you sometimes understand how things work. And today it's my pleasure to talk about understanding startup valuations and using a SMART Cap table. And we developed this at the Keiretsu Forum with 3 other investors. Because we were pondering what's our return on investment. And apparently, the return on investment is highly dependent on the exit value. It's highly dependent on the deal terms, and then we have usually not only a single round. And so, to get you some insight, as you will see, it's very easy because if you follow the formula that I'll present to you, you will quickly realize E = mc squared. And that still holds true today. And that's what I wanted to show to you. And so give you an understanding how to read a cap table and a pro forma cap table, how to eyeball your return on investment that you could have, and then get a sense which variables affect your ROI, and then have a dialogue with your entrepreneur on what is a fair pre-money valuation that makes everybody happy. The alignment of interest is really important. And I do think that the SMART Cap table that I'll show to you will give you a good tool to have a reasonable conversation with your startup founder. And I think that's really important to really see who am I investing in and how they are reacting. And so I would briefly talk about the cap table. The pro forma cap table is simple equity round. Then a little bit about, I will probably touch on dilutions and illusions. And then what a simple liquidation preference is. And then how pre-money valuations will affect the payout on a successful exit for the entrepreneurs as well as for you as an investor. So generally speaking, when we invest in companies, we often want to see a cap table, and because we want to know how much money we are going to make. And to keep things short, I think we have a total of 20 minutes for the presentation, 15 to 20 minutes, so that we have 5 to 10 minutes for Q&A. So to keep it short, on a cap table, what you only see is a distribution of ownership. And because you only see the distribution of ownership, you cannot know how much money you are going to make. So instead of that, it's often a question of what's your valuation and what's your exit. And so if you have, for example, a pre-money valuation of 5 million, the exit should be 500 million. Intuitively, the knee-jerk reflex that we would normally gravitate to would be to say, okay, could be 100x. Well, it actually isn't that simple, because if you raise 5 million on a 5 million pre, you actually end up with a 10 million post. You and the founder, or we investors and the founders, would have 50% each. The exit multiple would be a 50x, not 100x. Now, if the pre-money were not 5 million, were let's say 20 million, 4 times as high, you wouldn't have a 50x. You would expect a quarter, let's say 12.5x. But that's actually also not true, because at the end of the day, if you're raising 5 million on 20, you end up with 25 million posts, and you would have 20%. So your exit would not be a 12.5x, it would be a 20x. And unfortunately, not all startups end up with a 500 million exit. Some of them may actually only end up with a smaller exit. And if this is only 50 million, you end up with 2x. So it's actually, what this is dependent is actually not necessarily the pre-money. It's actually the post-money, because the amount, the capital that's raised also plays a role. And so your exit multiple is really dependent on your ownership percentage, which is basically your investment divided by the post-money, and that multiplied by the exit value divided by the post-money again. So, so this gives you a rough foundation on how to think about it. And now there is a little complication. The complication is, in most cases, this will not be the last round. There will be subsequent funding rounds that may or may not be at higher valuations. And so the question is, how can we think about it? What if we need 2 more funding rounds? How much will I be diluted? And to give you a rough rule of thumb, that's how I basically calculate it. Most investment rounds end up at about 20 to 30% of the post money to give that up. And that means it reduces your ownership down to 70 to 80%. And if you do this, and you do it twice, you have your ownership reduced by about 36 to 51%. So I usually, as a rule of thumb, if a company needs, let's say, 2 funding rounds, I usually think about a 50% dilution in terms of ownership. Now, what really matters is that the devil is in the details. It depends on how much capital do you need? How much capital will you need to raise? How will the valuation be at the next capital raise? It may, if you are investing in a note that has a discount or a cap, it may actually also depend on the discount and the cap. And so in that regard, those are 2 very important variables for bridge notes or convertible notes that we should always have. And so at the end of the day, what you want to see is sort of a table that spells out the shares and ownership, but also then what already money has been come in, what are the deal terms, and then what's the percentage of ownership after the Series A, after the Series B, or whatever series you have, so that when you have an exit, you get a sense what the payout will be, and that you can actually have a sense of what your exit multiple could be, which you see here on the lower right corner. And that is actually something that does require a little bit of assumptions. You see some of the fields are highlighted, and you need to go through the motion and punch in the numbers to get really a sense on where this would go and what would be the exit amount that one can expect. So with this, I actually would like to switch over to a Excel sheet that we developed, I think it's almost 2 years ago. And what we have done here is we have basically created such a table, and I hope that everybody can see it. And you can, what I should do is I should actually expand on this here. We usually put in the fields how many shares are there for the founder and the employees and contractors. Often we start off with 10 million shares. And then imagine there is a note, one is raising the very first note before it converts to the first series. And often they are called seed, and often they are now called pre-seed. But in principle, I called it Series A, B, and C. And if the note cap, let's say, is 10 million, that's what you see nowadays, 20% discount. They want to raise later on, let's say 4 million with a 12 million pre-money valuation. Then it's a share price of $1.20. And so the total investment would be 5 million. And then you would actually see what here the ownership would be. You would actually see that the note holders would have a, let's do this. This is easier to see. The note holder will have a 7% ownership, Series A 23%, employees, contractors 13%, etc. When then the next funding round comes, you basically have, and you want to raise 20 million on a 40 million, then there is, of course, a dilution, right? The founder gets diluted. The employees and contractors get diluted, and you could also actually put some numbers in here, but for the sake of simplicity, we leave that out. The note holder will be diluted, and also the Series A will be diluted because Series A will have something. But at the end of the day, you actually have a value increase. So it increased from 120 to 278. So the investment is then worth 2.9 instead of 1.25. So there is already a 2x plus multiple. So the dilution is in terms of voting rights, and it could be an illusion that you're losing money, because what matters is your value increase that would be if this would be an exit. And if there is then an exit, let's say here at 100 million, you can basically see what would be coming out of this. And I'll collapse this one here, those percentages. You will see that under these terms, you'll get a 5x multiple. That's not much. The founder, if everything goes well and there is an exit at 100 million, founder gets 37 million, and you go home with a 5x multiple while having taken all the risk. That might not make sense. One of the things that is often the case is, and I'll make it a little bit smaller to give you an idea here on what the multiples look like for the different investors here. Very often investors are asking for a liquidation preference that you see here. And what happens is, if the payout is they get paid first, they're at the liquidation, their payment will be preferred. And then the others will also want to have a liquidation preference. And so it looks already a little bit different. So the gray one is the entrepreneur, the payout basically on the pre-money that first started with the Series A. And then the other, the A would be in orange, which is over there. And then the B is the blue one. This is with normal liquidation preference. When you have participating preferred, that actually means that first it's paid out, and then you participate in that round again. And as you will see, what it actually does is it also changes the slopes. And as a result, there is a little bit more upside. And when you actually get paid out, you don't have a flat interval here. The challenge is at 100 million exit, this does not make sense. So one actually needs to look at the market size. And very often what we get snowed with is billion-dollar markets. But what is really important is what's there's this TAM, SAM, and TOM. We really need to understand what's the serviceable, obtainable market. And the serviceable, obtainable market will then, let's say, determine your exit value. And when you have here this exit value, let's say, and it's 250 million, then you're getting into a range that is actually more decent. What's interesting here is, and I'll make it a little bit bigger, I hope you can still read it. What is interesting is, if you do have a $250 million exit, the founder, you get a 12x, and the founder goes home with 83 million. That's a boatload of money. And so one of the things is that the valuations that are set here will actually, in a nonlinear fashion, affect what the founder gets out of this and what you get out of this in terms of the multiples that should actually justify the risk you're taking when you're investing in an early stage company. So for example, we have here 12x, 80 million on payout for the founder. If this 10 million valuation, and that's a very common number we see nowadays, if this would be just 5 million, what do you think? How much would the founder be diluted? Would he go down from 80 million to 40 million? And how much would the exit multiple increase? I think the exit multiple will likely, you will probably, that's intuitive, you will almost get twice as much. So let's see what happens here. So you have not exactly twice as much because there are some other downstream effects. But what's interesting is you have a 22x multiple instead of a 12. The founder gets 78 million instead of 83 million. For the founder, it really doesn't matter. And so this is the reason why such a tool is useful. You can show this to the founder, and whether you do it with participating preferred or without participating preferred. So this is without participating preferred. Let's switch this off. For the founder, it really doesn't matter much whether the valuation at the very early part is 5 million or 10 million. I switched back to 10 million. Now it's without participating preferred. You would get 12x, founder gets 92. If you have 5 million, you get 22x, founder gets 87. There's a very small difference for the founder when there is a reasonable valuation, but it rewards us as early stage investors, us as angels, for the risk we have taken, and it rewards us for the support and for the value that we have contributed to make this company a success. And so these are the numbers that I would think is important to play with and to become aware of it, so that you actually have a decent progression, and that everybody who is investing in this company will actually have a fair, I would like to say, exit multiple that's commensurate to the individual's risk. And we can here play with another numbers. What if you don't need the additional round? How will it affect it? Yeah, so this is kind of, I told you about 50% dilution with 2 rounds, so about 25% dilution with 1 round. So you see this is actually changing this accordingly. So you can actually punch in those numbers and see how this affects it. What if this valuation suddenly is almost a down round? So let's say it's only a 5 million, but subsequently you will still be able to raise this capital and have this exit, then you will still actually do relatively well. So as long as you actually have here a valuation cap and a discount, you actually have a huge benefit. So I go here with back to 12, and you can actually look here at the cap table, how the payouts will turn out and how the multiple will turn out based on the different exit values that you may have.
So we have 2 questions.
Okay.
The first question is, do you show the potential shares you may issue at the end of a convertible note on the cap table as reserve shares? So how do you show a convertible note before and after conversion on this spreadsheet?
Yeah, so that's actually an interesting thing. So we do have them here spelled out, and that assumes that there will be a conversion at this value. And if, for example, this is at 12 million, so in this case, it's a 5 million is here the valuation cap that kicks in here. So it's a lower, usually it's a lower of the 5 million or the 20%. And if this were, let's say, would be a 4 million, you would actually get proportionally more shares because now the cap doesn't kick in, but the 20% here kicks in. And so we have here basically down what you can do is a calculation on how many shares would be issued for the different notes. So you can have up to 3 notes in this tool that you can put in at different values.
Great. The second question is, do you make the spreadsheet available, and if so, how?
Yeah, so currently it's only an Excel sheet, and if you are an investor, I'm happy to send it to you. There is this was actually quite a major effort. There are a couple of hidden sheets behind it, and we actually would like to make this available on the web so that anybody can access it. I just haven't gotten around it, and it requires some bandwidth. So if anybody has some ideas, that would be great. But in principle, when I send this, I'm happy to share it with individual investors, and you should just contact me. And what's important is I'll send it out protected, and that is because there are a number of formulas here, and if you were typing stuff in here, you would actually destroy the formula. So you should also make sure that you only type stuff into the highlighted fields, and then it should actually work very well. I have now protected the sheet. I've done this without a password, so you can unprotect it anytime. The only reason why you would need to unprotect it is the scaling here of the graphs doesn't work when the sheet is protected. That's just the only limitation. So I would suggest use it as is. And if you ever want to see a different scaling on the graph, then you unprotect it and then do the new scaling, and then you protect it again, just to make sure that you're not destroying any formula.
Great. And we have one more question. In the current environment, what do you see as acceptable exit multiples for angel, seed, Series A, Series B, and Series C investors? What do you think would be good exit multiples at those stages?
Yeah, so I'm looking at the very first round. I'm looking at a 20 to 50x. I would like to see. So let's say a friends and family round. If you're raising 250, the 250 you can easily raise, and we can do this here. Let's do this. 0.25, and you can raise the 250 at, let's say, at 2.5 million. You do this for your friends and families. Then you raise the next, let's say, 1 million. And you do this at a, let's say, 4 million cap, and you do a 20% discount. And then you raise the next, let's say, 2.5 million, and you can put a 6 or 8 million cap on it. And what you can then see is what the different multiples is. So this is then the Series A with 12 million. And when you open this up, look what you, at a 250 exit, you still get your 60 million payout if you're an entrepreneur. But what you give to your first folks that come in, they have a 30x multiple, these have a 20x multiple, these have a 13x multiple. And so what I would suggest is stage it depending on how much capital you need to raise. When the capital that you need to raise is relatively small to the pre-money, the dilution effect is minimal, right? So here you have basically a 10% dilution. That's, or even, perhaps even, yeah, something like that. Here you have a 25%. And so at the end of the day, you want to stage it. And the advantage of this one is actually to get the money is relatively quickly. It smooths enough up your process, and you don't have to haggle with investors, because everybody nowadays with having an idea, often they start off with 10 million, and you have a huge advantage if you give a fair deal, and you only need initially for the next, let's say, value inflection point, a small amount of money. Stage it accordingly so that those people who support you at the very beginning, who took the highest risk, who made you successful, that they get rewarded for their support.
Great. And we had a comment from Dave. He says he can help with publishing this work. It's very similar to the topic in the Capital Access Series selection on valuation. So we'll make an introduction to you and Dave Mosby afterwards for that.
That would be great.
Cool. We have a few more minutes. What else would you like to talk about for cap tables?
So let's see. I have here our slide deck. So this is basically the summary here. So it's really, here we go. It's really essential to understand the implications, and it allows you to have an honest conversation with your investor, with your entrepreneur. So, for example, I had investors who were claiming, yeah, but my company has a much bigger exit value, so my pre-money is 80. And right out of the gate, because they said this is a more than $10 billion company. And well, that's pie in the sky. And if you use this table, you get a sense of who you are investing in. And at the end of the day, it's the person you are investing in. Are the interests aligned? Is this a person you would you trust and you would get on with, and who treats you honestly, and where we try to align our interests? I think it's this tool is useful also to get to know one another. And I even had entrepreneurs where I showed this table how to do this, and they realized their valuation was way too high. And they said, but we already have taken money in at that valuations. And I said, well, you can go back to them and say, in hindsight, we have actually done the recalculations, and we think you deserve more. And we think it would be, and investors are shocked. I mean, they're really appreciative when an entrepreneur says, well, I raised this at 8, but at the end of the day, the 200K I raised at 8, I can actually raise it at 4 or 2.5, and it doesn't make a huge difference to me, but it would actually, I know that this would actually make a big difference to you. So if an entrepreneur is even willing to give, in hindsight, the early investors even a better deal, that tells you something about the character of that person. So try to stay away from hyper-inflated high money pre-valuations, unless they are further along. Let's say they're close to market. They already have a 4 years history. They already have raised whatever 5 million or so, right? Then you're not, you cannot expect a 5 million pre. But in a very early stage, you want to have a good dialogue. So the key is really use this tool to have a honest conversation and see whether with this entrepreneur you can align interests.
So my last question before we jump to the presentations is, when you look at a cap table on a startup, what are the things you find as red flags? What jumps out at you as being a problem? If you had to pick, say, 3 of them, what comes up most often?
So if the entrepreneur if the valuations are too high and the entrepreneur hasn't been willing to give away a certain equity, so if the entrepreneur is too tight-fisted, one of the concerns are, and I'm not alone with that, is the whole concept on safes. That's because the safe is unsafe for an investor. It's basically a promise for a conversion, but you cannot insist on the conversion. The original safes didn't have a cap. The original safes didn't have interest, and it's neither a debt nor an equity instrument. So if the company goes under, you have nothing. So that's more related to deal terms. I do think that many investors, many entrepreneurs don't calculate ahead of time. So you want to see, has the investor thought this through to the end, and what will be left with you? You want to see the numbers, and very often the things are not very well arranged and sorted. So the red flag is, to me, if there are too many investments and the deal terms aren't clear, and the investor doesn't really understand how this all comes together.
Great, thanks. The audience also requested a copy of your slides, and what we'll do is share your email to have anybody who's interested can reach out to you to get the slides as well as the spreadsheet. It's a great presentation. I want to thank you for that today. And with that, let's go ahead and switch over to our first presenter, James Blackledge of Capella Imaging. James, love to hear more about your work and what you're doing there in the imaging marketplace.
All right, thanks all. Kind of a tough act to follow there. Thanks everybody, I appreciate the attention. I'm Jim Blackledge. I'm one of the founders of Capella Imaging here in St. Louis. We're a clinical stage startup that is developing nuclear imaging agents that are targeted at fibrin that is associated with blood clots. The group of us that founded the company have a great deal of experience in drug development and cardiology, and we're kind of excited about our lead candidate, Fibricent. So our main indication here is venous and arterial thromboembolism, or blood clots. And these are prevalent in the US especially. About 900,000 people annually present with some form of venous thrombosis. Heart disease is responsible for about 20% of the deaths in the US, and as heart disease has increased, the use of these mechanical support devices, whether they're ventricular.
James, you want to share your screen with us?
Oh, you're not seeing it? Oh, goodness gracious. I'm rambling.
We're getting a talking head, not bad,
Yeah.
but just may want to see the other part too.
There we go.
There we
You
go.
see it
Thanks.
now? Okay, yeah, the mechanical support devices, LVADs, prosthetic valves, these are becoming more and more common, and these patients are at an increased risk for some sort of thromboembolic event. And sudden death is the first symptom of a pulmonary embolism in 25% of the people. And my life has been touched by that twice just in the last year. So we need faster, more positive diagnostic tools for both measuring pulmonary embolism and for looking at the degree of thrombus accumulation in these mechanical devices. And so we have come up with this molecule Fibricent, which is a technetium labeled nuclear imaging agent that addresses these concerns. So this is a cartoon image of Fibricent. It's a tetramer. Each of the 4 arms is composed of polyethylene glycol terminated with 2 peptides, one of which binds to fibrin very tightly and specifically. It is a positive diagnosis of thrombus as opposed to a lot of the other diagnoses of PE, or you eliminate things. You say it's not this, it's not that. In addition, with some of the more acute concerns of thromboembolism, especially with the mechanical devices, there are currently no ways to do an assessment. The image you see on the right is taken with an LVAD. It's a left ventricular assist device, which is basically a pump that was removed from a patient that came in for pump exchange, and we imaged it with Fibricent, and you can see that very bright spot at the bottom of the rotating image is a thick layer of blood clot that has been plated out on the neck of this device. And this patient was at a severe risk of having a stroke because of that. A piece that could break off and go to the brain. So we have a low risk path to approval. Things I want to stress here is that most drugs fail in the transition from preclinical to clinical due to either efficacy or toxicology. Efficacy, it's the classic statement, we've cured cancer in mice many times over. It's not always effective to translate from an animal model to humans. In our case, as I showed before, that LVAD is our worst case scenario. That is an incredibly violent environment. There's massive shearing forces due to the pump. The patients are being treated with heavy levels of anticoagulant, but it is a human clot. We take those pumps out of a patient that comes in for exchange, we implant them in a 70-kilogram calf, and we acquire our images that way. So this is as close as you can get to clinical data in a preclinical model. The other thing is toxicology. As a nuclear imaging agent, we use exquisitely small amounts of material. And there's a correspondingly smaller risk of tox because of that. And the FDA actually acknowledges this. You're only required to do a single species of toxicology to get into humans. We have done this. We have a GLP tox study, and the report was completely clean. So this particular compound is extremely de-risked in both of these regards that are common for failure. So we're looking at a market that embodies really three primary indications initially. One is pulmonary embolism, and the second would be prosthetic valves, and the third would be the ventricular assist devices. And these all have varying degrees of needs for support, but we estimate that the sum total is going to be about 400 plus images per year for both acute and preventative diagnosis. And altogether, this represents a market of about $615 million. The valve market is expected to grow significantly, as well as the LVAD market. So it is a growing market, despite the fact that it's not good news if you're a human. It's potentially good news for our product. So our competitors in this area, in terms of ventricular assist devices, there's nothing. It's a completely unmet need. You're not able to use any of the common imaging modalities because you've got this, this very large chunk of metal in your chest. Prosthetic valves, we're up against the current standard of care, which are a couple of imaging modalities here. They're all some form of echocardiography. That works reasonably well, except that there's a great deal of image manipulation and examination required. If the first scan suggests that thrombus is present, it typically requires supporting images from other modalities to get a positive confirmation. No definite diagnosis until you actually do surgery. And one of the biggest attractions of this is common with valves there are two problems. One is thrombus, and the other is something called pannus, which is a fibrous growth of tissue over the valve, which is very different from thrombus. And the reason that's important is current imaging modalities cannot distinguish between the two. And the treatment is vastly different. Thrombus you can treat with various anticoagulants. Pannus requires surgery. In the case of pulmonary embolism, current standard of care is computed tomography and perfusion ventilation scintigraphy. We really see ourselves in the position initially of replacing the VQ scintigraphy. This is for patients that, for whatever reason, are not candidates for CTA. And VQ scintigraphy is very often indeterminate. You just can't get an answer with it. Whereas with our compound, you get a very bright spot which shows you exactly where the embolism is. We have key opinion leaders that think if all goes well and we have the specificity and sensitivity that we believe we do, that in fact this could become the first pass diagnostic treatment for PE, which would be a wonderful thing. So intellectual property. This was developed in the laboratory of Greg Lanza, who was one of our founders at the Washington University School of Medicine. Washington University owns the property rights. We have the exclusive license for this, and we've already worked with the GMP manufacturer to develop a scalable process for making this to support trials. And in that process, we made some changes to the overall structure that make it much more possible to make this on scale that we will eventually use to extend our patent protection. This is the development team, Dakota colleague. We have one great guy and a bunch of schmucks. But collectively, we've got a lot of experience. But these are the guys I really want to stress. We have a world-class team of medical advisors. Barry Siegel, Sally Schwartz. I was just at the meeting of the Society of Nuclear Imaging and Molecular Imaging and Nuclear Medicine last weekend. And both of these people have symposia there that are named after them in their honor. Sally wrote a lot of the guidance documents for the FDA around nuclear imaging. Greg Ewald is the chair of the heart transplant and failure group. Alan Zaharias is a big name in prosthetic valves. So we have fantastic medical advisors. We're looking to get a $150 million convertible note to get us through to IND submission and approval. After that, we will look to raise $3.5 million that will drive us through our phase 1 clinical trials. This is based on a initial $10 million valuation, followed by a $1 million investment by North Star Medical Radioisotopes. And that drove all of our preclinical activities to get us to where we are ready to go into the clinic. And as I mentioned before, we've contracted with a very good GMP manufacturer, but even more critical is we've contracted with an outstanding group that is very deep in nuclear imaging for managing our clinical trials. And these are just some examples. I'm still currently seeking the details of these deals, but you know, there are people out there, larger healthcare companies that are interested in acquiring. These are all nuclear imaging companies that have been acquired recently. So this is the timeline of what we're looking to do. You know, the $1.5 million raise we're looking to do now, submit an IND, clinical trial phase 1. We are looking to ultimately submit an NDA and get approval in early 2027 and looking to exit then through acquisition. And I will say that North Star Medical Radioisotopes, that's the whole reason they got involved. They're interested in the asset. They are a technetium generating company. And I touched on most of this already. This is just what we've done with the $1 million we already got. Made a lot of progress, all of our preclinical activities, and we're ready to go. So thank you.
Great. Thanks, James. And Chris, what questions did you have there or feedback?
Well, first of all, what I like about this technology is that it's a positive signal. Apparently, you can actually, it's not a diagnosis by exclusion, which usually is the case when we are looking at contrast agents. There is, however, with a CT or MRI, you can actually make a statement on the composition of this matter. So you can actually pretty definitively say whether it's a thrombus or not. The whole thing, the question to me is, how much easier accessible is it than, let's say, a low dose spiral CT, where you can run through this in 2 minutes and you may, you inject a little bit of contrast agent, and it's a fast procedure that gives you also other resolution and other information. Where I'm struggling, where is the differentiator?
No, that's a very good question. And I mean, we addressed the first part of it first, which is, you know, theranostics is a very, very sexy term now, and using a lot of exotic radioisotopes with really short half-lives and very, very rare. I want to stress that this uses technetium, which is already available at any hospital. It's a catalog item with Cardinal Health, for instance. And you're right, spiral CT works very well. You'll notice I didn't talk a lot about deep vein thrombosis, even though that's a huge part of you know, the embolism market. It's not really an unmet need. I don't see us right now as really going up against spiral CT. I see us going up as an alternative to the people that are, for whatever reason, that is contraindicated, whether they have issues with the contrast agent. You know, this is the perfusion ventilation scintigraphy population that is a subset of the PE patient population that we think we could be a player there. But it's in 2 minutes. No, it's not a 2-minute scan. I think 2 minutes is probably a bit optimistic for a spiral CT anyhow. But this is something that can be done on an acute basis in an emergency room. A patient presents with chest pain, you know, the technetium is always there. The Molly cows, as they call them, are always being milked in the nuclear pharmacies. And this is something that would just be stored in the lyophilized form and again, be prepared in about 20 minutes.
I was wondering whether you can argue with sensitivity. Is your method much more sensitive than other approaches? And the reason why I'm saying this is, so for example, in international flights, if you fly economy, let's say the risk for a DVT is probably somewhere in 1 to 1,000 to 1 to 5,000. it's not immaterial. And it's because there are many people who have coagulation issues. You may have an inherited protein S, protein C deficiency that you're not aware of because nobody tests for it. And you may actually experience a DVT after intercontinental flight, and you may not be aware of the risk of a pulmonary embolism. Now, most DVTs, I would think, that occur are actually subclinical. You have some swollen legs and you walk around for a couple of days and it's gone, or it's gone after a day, right? So most are subclinical, but the question to me is, could your tool be a more sensitive tool that's easier accessible to detect subclinical DVTs that otherwise wouldn't be detected?
So we are certainly much more sensitive than spiral CT and ultrasound that's typically used for DVT. And you touched on an interesting idea. I mean, that's another place where we think we could be a significant player is smaller peripheral clots in PE. And I should also mention that in a lot of the work with prosthetic valves, for instance, these are asymptomatic patients. They have no idea that they have these buildups. You know, when you start showing symptoms, a lot of times you're already in danger. And so this sort of high risk population, we see this as a big market. Is, you know, they should be screened every 6 months and evaluate the effect of your anticoagulants and things like that.
Great questions, guys. We're at the end of our time, but appreciate it. Hope we can make follow-up introductions afterwards if interested. With that, let's move to Jeff Carter. Jeff of Scaffold Therapeutics, can you turn on your slides and show us more about your technology there?
Absolutely. Thanks, everyone. It's nice to get an opportunity to present to you today. So today I'll tell you about Scaffold Therapeutics. We're a small molecule company targeting cellular stress mechanisms. So cellular stress is sort of an underlying disease driver in diseases across almost every therapeutic area. And it's a pretty active area of investment in biotech today. Of course, with biotech's tendency to not really be totally current on technology, I feel like it's important to put front and center that, you know, we're aggressively deploying generative AI platforms into our research programs, and they actually move things forward significantly. We've got a platform deployed across all of our discovery activities right now, and we've got development ongoing for translational biomarker identification and disease specificity. So, you know, keeping that in mind as one of the reasons why we've been able to iterate so quickly on our molecules, and then putting it in context of cellular stress. A moderate cellular stress response is really important for your body, right? So if your cellular stress response is too low, which it weakens over time as you age, you can over time end up with some of these classic neurological diseases of aging, dementia-related diseases. And so we're looking hard at that. These are incredibly expensive drugs to get onto the market. So our path there is to partner that program out. And I'll talk with you about that as we move through the slides. But for a small company like ours, we can actually move an oncology molecule into the clinic in a tractable amount of time and on a tractable budget. So in parallel with that, we're really looking at liver cancer and a number of other malignancies. And keeping to the story on cellular stress, you know, a cell that exhibits a very strong stress response is able to actually fight off the body's natural immune system when it's sort of bouncing around in your body. And when these cells clump together, you get these aggressively growing tumors. And a lot of that is moderated by these cells' ability to really generate a very robust cellular stress response. So how did we get to talking about these two very different indications at the same time? We followed the data of the compounds that we've been developing. We originally in-licensed the compound, and then we've taken it several iterations since then. So in neurodegeneration, there's a demonstrated mechanism for amyloid beta clearance associated with our gene target. So the gene that we're reducing the activity of is called metal-3. Our compounds also readily penetrate the brain even after a single dose in a live animal. And that's a really key differentiator that we'll talk about when we cover the competitive landscape. Now in liver cancer, it's a little bit different. The first thing to know about metal-3 as a gene that we target is that it's everywhere in your body. So what we need to make sure of is that we don't too heavily reduce the activity of metal-3 in healthy tissues. We really focus on going after the disease tissues. So you can see on the bottom right where the compound goes in an animal, right? Not much of it goes to the plasma. Some of it goes to the spleen, which is pretty normal. And then most people think, well, yeah, everything goes to the liver. That's not really true. Everything goes through the liver, right? The job of your liver is not to absorb everything. It's to sequester it and then excrete it, get it out of your body. Our compounds are actually taken up by the liver, penetrate the tissue, and accumulate it. And the fact that metal-3 is strongly correlated with with liver cancer, and also strongly correlated with negative outcomes in humans in liver cancer, tells us that we have a shot at moderating disease or ameliorating disease in the liver, and not hugely impacting these other tissues. So how do we know that? Well, we look at academic and published data, and these are academic experiments where the metal-3 gene is just turned completely off in an animal. We can't do that with a drug, and so I'll talk to you about what that means as we kind of go through this slide. But on the left-hand side, in mouse models of neurodegeneration, you can see the black bar, significant buildup of amyloid beta plaques in these mice. These are normal mice. And then when you silence that metal-3 gene, turn it off completely, those amyloid beta plaques are cleared very aggressively and reduced down to a very low level. And you see the same thing in liver cancer. So you take a so-called wild type mouse, and you inject it with a liver tumor, and it grows very aggressively with a normal level of metal-3. You silence metal-3 in that mouse, and you end up with the bottom right image, which is those tumors are dissipated very quickly, and some of them disappear completely. We're running an efficacy study with our molecules in liver cancer right now. So we do have robust IP for everything that we're doing. Our lead program, we licensed from the University of Zurich. We really like the work that they're doing on this set of targets that we're investigating. And so we also have multiple sponsored research programs going on with that lab. And the subsequent patents that have come out are co-owned by Scaffold and co-invented by Scaffold. And the balance of this year will be filing our own independent IP, both in the technology space and also in the chemical matter space. So there's a lot of activity right now in cellular stress. You can see the industry activity at the top. These are some of our top targets for partnering of our neurodegeneration program. We have active conversations. By active, I mean multiple communications per week with some of these companies going on right now, with a target to close a partnership this year. A lot of venture capital money is coming into companies in the space, but it's still not too crowded, which I'll talk to you about on the next slide. We're working with a lot of the top tier academic institutions. We're working with all these institutions at one level or another, whether it's through our SAB or otherwise. And of course, like everyone, we're aggressively applying for as much non-dilutive grant funding as we can get. So in terms of the landscape, we know that our competitors' molecules target this gene effectively, but do not cross the blood-brain barrier and enter the brain. And so for this partnership initiative that we have on the neurodegeneration side, we feel like we have a wide open net, at least until the end of the year. So the pressure's on me to get that license closed and do something interesting in a partnership with neurology while that opportunity is wide open. In oncology, there is some competition, but for the amount of money that's come into the space, not a whole lot. And especially if you narrow it down to small molecule, which is what we're doing. Storm Therapeutics has a phase 1 study going on right now. mostly in liquid tumors. Most of their data has been in lymphoma and leukemia. 858 Therapeutics is about at the same stage as we are in terms of development, and most of their data have been published on colorectal cancer. So we feel like, especially based on the data that I've presented in the talk, that we have a nice shot in liver cancer. We think our compounds naturally get us an opportunity there. I'm a repeat entrepreneur. I built and sold a company called Arcspan, which was a multi-tenant SaaS company, to Bruker, which is a life science equipment provider. It's a publicly traded NASDAQ company. Did that in 2019. Gordon Saxty, head of chemistry. He's put drugs on the market. Balversa is a blockbuster J&J drug for metastatic bladder cancer. Jeff Hickson is a dyed-in-the-wool biotech operations guy and a biochemist who makes sure that we get the best deal for every dollar that we possibly can on the operations side. And then Wasim Malik led our last 2 rounds and will be leading this round as well as part of Yasso Ventures. And we've got the best of the best in terms of advisors, including Kathy Burns, who's the chair of pathology at the Dana-Farber Cancer Institute. So in terms of our round, and cognizant of what Dr. Apfel presented earlier, we do have a safe at a $15 million pre-money valuation cap open now with a 20% discount. I've collected 15% of that money, 25% committed, and I've got a lot of open interest. In parallel with that, we can offer a convertible, so let me know if there's interest in that. In terms of business goals, we'll close that first partnership. We're going to be recruiting a CSO. We have a couple of finalist candidates already and get some grant funding. On the product side, a development candidate in liver cancer. We've got to run that efficacy study in Alzheimer's, and we've already started bringing that second pipeline asset up. You can learn a little bit more about what that is on our website. We've raised $2.2 million in safe funding already, and we burn, it's really about 100K per month, but I round everything up. So our burn rate is a little bit lower than what I've documented. We think we have visibility to a $200 million exit in 5 years. You can see that I've got rounds laid out there that bring us to about a $25 million total amount raised. These 2 comps are very similar to what we're doing. The difference in valuation is market conditions. Neither of them were generating revenue when they exited. Constellation exited in 2021. Epizyme exited in 2022. So my projection is at the low end of the 2022 range. There's no market going up built into my projections, so I feel like it's a good baseline. So anyway, in closing, we have great IP. We've got a really hungry team, and we're excited to keep achieving our next stage milestones. I'll take any questions.
Great, thanks, Jeff. Chris, what did you think about that one, and what questions or feedback did you have?
I would like to know how it works, what is there, and how much capital you have raised. One needs to actually run the numbers through the SMART Cap table. As I mentioned, so safe is not safe for investors. So it's not really in keeping with what I said. The pre-money looks pretty high. So the question is, what would be the ROI? But you started off with saying that you're using machine learning and AI in order to develop the drugs. I didn't understand. I mean, it's a very active field to use this technology for drug development. I don't know what the success rate has been to use AI for the drug development. My understanding was that it has been in the past fairly low. It hasn't really kept up with the promise. But I would love to understand, what AI technologies have you used? Are these simple artificial neural networks? Are these convolutional neural networks? Are we talking? So that would be of interest for me to understand. And then the other question would be, what has been developed, and are there preliminary data, and what do they suggest? Is there IP? I saw one compound, so that it sounds like you have a composition of matter IP, And then the question is, how do we know that this will work? You mentioned that very often, I think it was you mentioned that if you have things in a mouse model, you may be able to show it. Cure cancer in the mouse model is easy, but curing it in the patient is difficult. It's actually not necessarily the mouse model. It's actually the tumor tissue that you're looking at. If you're looking at cell lines, that's easy because we know the cell lines are sensitive to this and that. Whether you test it in vitro or in the mouse, basically gives the same result. If you take a PDX model, a patient-derived xenograft model, in the majority of cases, you're not curing this mouse. and so mouse model is not equal mouse model. It's really on what tissue are you actually looking at? Are you looking at the cell lines, which most of the cancer researchers do, or are you looking at real patient tissue, which is biologically very different? But apart from that, if you can allude to what AI are you using and what has this revealed? What was the data? What's the composition of matter? What's the evidence that this will work, and how should this work? So you can certainly answer that in 10 seconds.
I'll try to do what I can. So we're using a large language model that's protein specific. It is a published model, but we're training it with our own structural enablement data, X-ray structures. And primarily we're using it to identify allosteric sites, so non-enzymatic sites on the proteins that we're trying to drug, which tends to be pretty high value. The first drug that was designed with AI just went into the clinic this month being July. So that's it. So it is a rapidly evolving field. In terms of, we are using cell lines as this step. There's a new technology called mini PDXs where you can test human tumors in mice. That will be our next step. It's included in the budget for this round. I'm over time, so I'll take a pause there, and hopefully that covered at least a little bit. IP, we're very well protected across multiple chemical series.
You can finish your thought out there, Jeff, one more minute, and then we'll probably need to move to a follow-up meeting to cover the rest of it. But if you had any further thoughts, go ahead and play it out one more minute.
I think, let's see. I think those were the high points. I think I won't press my luck.
Okay, great. Well, thank you, Jeff, for the presentation. Thank you, Dr. Apfel, for all the great feedback and the great presentation on SMART cap tables and so forth. We'll make that available to all the audiences, your contact details, so they can get back in touch with you for the slides and the spreadsheet and anything you offer with that as well. And James, thank you for your presentation. Capella was really great. So with that, I think we'll go ahead and wrap up for the day. Appreciate everybody in the audience asking great questions, and we'll see you guys next time. And with that, we'll close it out.